Hi everyone, my name is Chinkai. Knowing which cell types a gene is expressed in has always been used as a clue for gene function. What I'm going to talk to you about today is how this basic type of knowledge is the main reason why bulk co-expression data is informative about gene function. First, a little background. Functional genomics has always been an active area of research, and up to now, the characterization of functions of many genes are still incomplete and remains to be a challenging task, especially many novel genomic elements. Some non-coding genes are continually being discovered, so people have developed many models which take as input many different modalities of information, including DNA sequences, protein structure, and expression profiles. Among these methods, co-expression analysis has been used in many functional prediction algorithms and workflows. It is well established in the literature. So what is co-expression? It refers to the coordinated variation of gene expression levels among samples in a transcriptomic data. So when using co-expression networks for gene function prediction, a central idea is the GBA principle, which states that co-expressed genes are more likely to be functionally related than genes which are not, which means functions of well-studied genes can be easily propagated to their co-expressed but poorly annotated genes. Despite its widespread use, the specific reason why co-expression has predictive value are surprisingly poorly delineated. It is usually assumed that genes which are co-expressed collaborate in a biological pathway or process. This might be true for some genes, especially those whose products from parts of the protein complexes or transcription factors regulate their target genes. However, given the fact that most studies build co-expression networks from bulk tissue transcriptomic datasets, which contains the contribution of multiple cell types, it is reasonable to assume that cellular composition can somehow affect the predictive power of such co-expression networks. For convenience, we also defined a term called cell type profile, which simply means the expression levels of genes across different cell types. Previous studies in our lab have confirmed that cellular composition accounts for a lot of variance in bulk transcriptomic data. As shown in this schematic, this is a toy example where we only have two cell types and a bunch of genes. If you look at gene 1 and gene 2, so they have the same expression patterns, that is, they are only expressed in cell type B, and the proportions of these two cell types vary across different samples. So the result is that the gene 1 and gene 2 will be highly co-expressed in the co-expression network. So basically, this study reveals that strong correlations between genes do not necessarily indicate a regulatory relationship. It can simply because these genes have consistent expression patterns across different cell types. This study also confirms that gene modules identified in co-expression networks are often enriched for cell type markers. So now the question is, where is the predictive power of co-expression network coming from? We hypothesize that co-expression network built from bulk RNA-seq data can be used for function prediction, mainly because the cell type profile information embedded in it. Based on our previous work, because cellular composition is a major contributor to co-expression analysis in an unsupervised setting, it makes sense to directly test whether the same phenomenon explains predictability in this supervised setting. So now let's take a look at the pipeline I used for function prediction based on bulk RNA-seq data. Basically, we used a GBA-based classifier which takes two inputs, the co-expression matrix built from the GTEx bulk data, which contains 54 human tissue types, and a binary label matrix obtained from gene ontology. so we only kept the Go terms from the BP category, which leads to around 3,000 Go terms. And I will use the word Go term and gene function interchangeably. So the output is a probability matrix indicating whether a gene is likely to have certain functions. Then we can compare the output with the ground truth, so we can evaluate the performance for each gene and for each Go term. So specifically, I used area under curve AUC as a performance metric. So here is the result. So each dot represents a tissue type here. And this is the maximum AUC across all tissue types. So we can see like the value, even though it's not very high, but it is comparable to previous studies based on co-expression analysis. Particularly, we looked at the distribution of AUC for brain cortex. So in the background is a null distribution given by a dummy classifier, which just randomly assign functions to genes. So it's just around 0.5. Overall, all the above results demonstrated that we can successfully recapitulate the basic observation that co-expression has some information on gene function. Obviously, this works because groups of genes sharing a functional annotation are more co-expressed with each other than with other genes. So I used the silhouette coefficient to measure whether the member genes of a Go term are strongly co-expressed with each other. So as you can see, it is positively correlated with the prediction performance. Next, I'm going to dive deeper to explain the source of predictive power. I used two independent but complementary approaches to demonstrate that predictability is largely explained by cellular composition variation. So I obtained a single nucleus dataset from Allen Brain Institute. With this dataset, we can generate simulated box samples while the cellular composition can be artificially manipulated. So basically, some cells from each cell types are randomly drawn from the dataset, and their expression profiles are averaged and weighted by corresponding proportions. So we used these baseline proportions, and based on normal distribution, we can generate random numbers, and we used a parameter to control the level of cellular composition variation that we want to introduce. So the results clearly show that upon the introduction of cellular composition variation, the prediction performance improved. To rule out the possible confounding factor, we also repeated the same analysis using some other randomly generated initial cell type proportions. What we can find here is that the prediction performance is only affected by the amount of cellular composition variation and is insensitive to the exact choice of initial proportions. So this result is highly reproducible. We also compared the results from the simulated dataset with the results from the real GTEx box datasets, and we can see there's a clear correlation between them, which confirms the fidelity of the simulation. Next, the second approach, we were trying to identify if there are subsets of samples in the real box dataset that naturally have similar cellular compositions. So we expect if we just take out these homogeneous samples and make a prediction, it will lead to a lower performance due to the lack of cellular composition variation. So we considered several deconvolution tools. So specifically, we used SurveySortX. So it will output the estimated cellular proportions. We performed a hierarchical clustering using the estimated cell type proportion and identified several homogeneous clusters. Here are a few examples. Compared to random sampling, all the samples within a homogeneous cluster tend to have similar cell type proportion. That is to say, there are much lower cellular composition variation. We were also aware of the fact that prediction performance is also affected by the sample size. So to obtain the baseline performance, we used a bootstrapping method, and finally we can use a polynomial model to fit the relationship between them. As a negative control, we randomly selected some genes, we call them fake markers. This is to deliberately make the cell type proportion estimation inaccurate, so the resulting clusters are still highly heterogeneous and their performance should still be very high. The result is exactly as expected. Comparing to the baseline, all the homogeneous clusters end up with much lower AUC value. Overall, the result supports the idea that cellular composition variation is essential for function prediction. Next, I will show you how predictability is contributed by the corresponding cell types. This is the result from the simulation analysis. Among the highly predictable goal terms, lots of them relate to immune responses. We hypothesized this is due to the existence of microglia cell in the brain. So next, we just removed this cell type during the simulation. And from the result, we can see that most of these immune-related goal terms just disappeared, while the goal terms that obviously involving other cell types are largely not affected. We conducted the same removal experiment for all the other brain cell types. By comparing the AUC values before and after the removal, we can tell whether a goal term is highly responsive to a specific cell type. Next, we looked at the expression profiles of mammal genes in these highly responsive goal terms. And here are a few examples. So in most cases, what we can find is that if the performance of a goal term is highly affected by the removal of a certain cell type, it happens to be that their mammal genes are just highly expressed in that particular cell type. So now it is clear that if you remove that cell type, you will destroy the expression patterns, and then you will just make that goal term no longer predictable. We used silhouette coefficient to measure whether mammal genes form a goal term or have consistent cell type profiles and find that it is correlated with the AUC value, which means cell type profile is important for function prediction. Finally, we directly make prediction, just use cell type profiles, and find that the results are highly correlated with co-expression network-based prediction. To wrap it up, cellular composition is an important source of predictive power in co-expression networks built from bulk data. If there is enough amount of cellular composition variation and all the genes have consistent cell type profiles, it will make the corresponding functions more predictable. Cell type profiles can be directly utilized for function prediction, but this method can only work if the functional term has something to do with cell types. So don't expect this method to work for arbitrary functions. Besides, according to our observation, even for some of these housekeeping genes, like those involving ribosome and protein synthesis, can still be predicted using cell type profile. This is possibly due to some expression patterns in the cell type profile. For example, some cell types might be more metabolically active relatively. So anyway, knowing which cell type a gene is expressed in and at which levels relative to other cell types is informative about its function. So we can simply look at cell type profiles. Of course, there are many other sources of information, but we hope that by making this particular type of functional clue explicit, we can improve the interpretability of computational methods for prediction. The above analyses are mainly based on brain tissues given the fact that there are lots of publicly available datasets. Repeating the same analysis on other tissue types might reveal whether the phenomenon is universal and whether the conclusion is broadly applicable. Cell type profile-based prediction itself might not be highly competitive but it can be easily integrated into other pipelines or workflows. In ensemble learning, people can design multiple models based on different algorithms and using different types of inputs and combine them all together, and it typically will lead to a stronger performance. And now it is possible to obtain high quality cell type profiles with different levels of granularity and from different conditions with the advancement of single cell sequencing. So it will provide a comprehensive information and help improve the prediction. So finally, I would like to thank my supervisor, Dr. Paul Pavlidis, and all the amazing lab members. Without their support and help, this work would not have been possible. Thank you.